Welcome back, everyone. Next, we have Regen BioPharma Inc., trades on the OTC Pink sheets under the symbol RGBP, is a biotech company focused on developing innovative treatments using autologous cell therapies, RNA and DNA-based immunotherapy, and small molecules in the immune-oncology and autoimmune disease space. Please welcome David Koos, President and CEO, as well as Harry Lander, Senior Scientific Consultant, as well as Dinesh Singh, Scientific Consultant. Welcome, gentlemen.
Well, thank you very much. Good to be back.
Always happy to have you back. David, kick it off.
Okay. So today, two major things we want to cover. We have the results from our recent study on Duracar, and then we have some further information on our moving forward on HemAccelerate, which we've cleared an IND on, and we're looking to move that into the clinic. So to start off with, I think we'll have Dinesh talk about HemAccelerate, and I'll hand it off to you.
Hello.
Hi, Dinesh. Yeah.
Good afternoon, everyone. So I'm going to present about the status update about the two different programs. And so the talk is divided into two different parts. The first part is related to HemAccelerate, as David mentioned, followed by updates on the Duracar, which are our Senior Scientific Consultant, Harry Lander, will update you. So without further ado, I'll just take you through the slides. So this is your safe harbor slide, just to be so that all of you are aware of the risks involved in any biotech company that matter. And just take a moment to glance through it. Next slide. Well, as you must be aware, so we have a lot of portfolios and a lot of programs under our umbrella. However, for the purpose of today's talk, we're going to focus on two of the programs briefly highlighted by David. The first one is the HemAccelerate, which has been cleared by the FDA for the IND. So the IND clearance for this program is done. So this is a drug product for anaplastic anemia, and we are all geared up to take it to the next level. The second program is related to Duracar, for which Harry Lander will give you the recent data updates that we have got. So just to walk you through what is HemAccelerate, what it is for, and how we're going to proceed, I'll just slowly take you there. So in terms of the incidence and the outcome for the anaplastic anemia, we know that one or two people per million people in the US alone are diagnosed for the anaplastic anemia, or aplastic anemia, that's the other name for this. So in theory, there are about 600 diagnoses per year. And because of the rarity of this disease, this comes under the category of orphan indication. So this is the orphan disease for which there is some cure, but however, a lot more needs to be done. So globally, however, in terms of the population size, there are around 48,000 people who are diagnosed and are still there. But that does not include the people who die of these diseases because of some other complications associated with it. So in terms of the state-of-the-art treatment for this disease, we have the bone marrow transplantation. However, because this disease, and remember that this disease can happen at any age and irrespective of the gender. However, the 5-year survival rate for this wanes with age. Not only that, you have the graft versus host complications and the associated toxicities thereby. So, and also in terms of the response rate for the patients who are treated, there are only less than 50% of them which responds properly, and they have to be on the immunosuppressive. For the patients who are already have gone through the bone marrow transplantation, 30 to 40% of the responder, 30 to 40% of the responders basically have a relapse of the disease. So in summary, I just want to draw your attention to the fact that though there is a treatment available, but there are patients who don't respond to this, and they are in urgent need of an alternative. So that is where we come in as Regen BioPharma with our adipose-derived cells, which we call the Hem Accelerate. And one of the advantages of having this drug product, this is a cell-based product. So one of the advantages for this is that it is simple and non-invasive procedure is involved in terms of harvesting the cells. This does not involve the bone marrow or so. Because there is a high content of the mesenchymal stem cells in the isolated tissue, so it not only takes care of the cells that are needed, but also provide niche for the proper development of the cells in the bone marrow. Also, because this is derived from adipose tissue, and the number of mesenchymal stem cells does not decrease with age, as is the case for the bone marrow stem cells or the hematopoietic stem cells. So this is a big plus, irrespective of the age, when we're talking about the function decreasing with age. Recent studies have suggested over the past 5 years, people have done using single cell analysis of the tissue heterogeneity. They have mapped it both in the patient population and different disease conditions, and even the normal individuals. And what they have found is that the cells derived from the adipose tissue consists of at least 60 different types of cells. Here, we're just listing, say, for example, the endothelial cells, the fibroblasts, the adipogenic cells, and immune cells. Even in the immune cell category, we have the macrophages, T cells, NKT cells, and several types of them as well. In just the point I want to draw attention to is that this is such a rich niche that we have so many types of cells which could be put to different uses, and it basically reconstitutes the whole system. And the fact that it is also derived is autologous from the same patient that we're putting it in is an advantage, that we have very less toxicity effect, and the safety is already taken care of. So,
Dinesh, let me interject a question real quick.
Sure.
So, you mentioned at first about 600 patients on an annual basis get aplastic anemia. Mm-hmm. Now, to the average person, that may seem like a small number, but the one thing that it doesn't say is that with the problems derived from chemotherapy, that
absolutely. So this is this Hem Accelerate is a wonderful drug that it could not only be used as an aplastic anemia, which is the population size is small, but also in cancer patients where the population diversity is very huge, especially in chemotherapy-induced neutropenia, and NK cell absence, Not only that, these cells could be isolated, prior to the patient seeing any chemo or drug, and could be reconstituted for, giving them to avoid the toxicity and complications in cancer. Yes, but as a proof of the concept, what we are interested in is we're using it in aplastic anemia. But the potential is huge for this drug. And given the fact that there are 60 different cell types that are found, this is an opportunity to extend the fact that we can isolate the specific cell types, modify them in vitro, and put it back in the patient. So that is the next step that we will be doing and exploring and having more INDs. They're all planned out, but just as for this audience, as a proof of the concept, we are focusing on the aplastic anemia because that is what we have been cleared for by the FDA right now.
Well, the going through the whole aplastic anemia, it seems like we're laying the foundation so that we can prove, first of all, it's not toxic to patients, and it does have very therapeutic benefits to it. Then we can extend on once we've got that foundation laid. Is that a fair statement?
Absolutely.
Excellent.
So now, go ahead, please.
No, go ahead.
So now, so this product is supported by our IND. So we have a patent related to this, and the two main take-home messages from this patent is that we have a composition and method for accelerating the bone marrow reconstitution. Remember that aplastic anemia can happen in two conditions. One, when you don't have these hematopoietic stem cells in the first place, or if the hematopoietic stem cells are not functioning properly because of some deficiency of the niche. So both of these could be taken care of by Hem Accelerate. So any patient who are refractory to the bone marrow transplantation or not responding to can very well respond to with a safe treatment that we have with Hem Accelerate. So we also have the protocol for generating the stem cells, for this. And here we're mentioning including aplastic anemia, as David mentioned. This could well be used even in cancer patients and some other autoimmune disorders and other things. The key there is in those indications, we'll have to isolate different specific types of cells. Since the markers and all the things have already been mapped out, it would be a cakewalk for us for the next level. So now, talking in terms of the procedure, I'll just briefly go through. It's so simple, that I have no words to describe it. So we have a fatty tissue, we lipoaspirate it, enzymatically digest them to get rid of the fat and all the debris. Now we have a pellets of cells. And these cells is what we put back in the patients. Some people might ask, oh, why would this work in the first place? So you can understand it in simplistic term or layman terms, that it's just like rebooting your computer, that you still have all the components, all the hardware and everything. Many times your computer crash, but then the moment you reboot it, everything becomes fine. So even in case of patients who are not responding in case of aplastic anemia or even in chemotherapy, right, just taking these cells out from this adipose tissue, purifying them, putting it back in the same patient, make them work, and there's a high potential. We have high confidence to believe that it will be a great success. So now moving back to give you a clinical synopsis of the things we have in place. So as I mentioned, the investigational drug product that we call Hem Accelerate is basically an autologous that is taken from the same patient, adipose stromal vascular fraction of cells. And each word is important here because, and it is self-explanatory in the sense that it is derived from the adipose tissue, it's from the stroma of the cells. They are enriched in endothelial cells and other supporting cells and so on and so forth. So this, just to make it compact, it's a self-explanatory term that we have, and we have codenamed this as Hem Accelerate. In terms of the study that we have submitted to the FDA, the title goes like autologous adipose stromal vascular fraction of cells for severe immune-suppressive refractory aplastic anemia. So even in the aplastic anemia category, we are right now, just to demonstrate the proof of the concept that how powerful our technology is or how drug product is, we are trying to enroll patients who are refractory or are on immunosuppressive drugs and refractory to the bone marrow transplantation. And the time that we will take to complete this study is only 6 months after the last patient has entered the study. And here are the details about the study objective and the dosing, because this has been gone into the phase 1, is going to phase 1 and 2. So as you understand, it is mostly about the safety and the efficacy demonstration. So the safety part is very easy, and so is the toxicity part, because there are other results people have shown, taking adipose-derived tissue for other diseases or indications where they have gone up to 150 million. Here in our case, we are going up to 100 million cells per patient in a plastic condition. And we have no reason to believe that it won't be a great success. I'm very hopeful about it. So as David mentioned earlier, so in terms of the market potential for the drug, so the chemotherapy-associated neutropenia is about a $13 billion market from last year. So is the hematopoietic, about 2.56 million, and radiation protection from government procurement about 1.5. So we are looking at anything between 15 to 16 billion drug product market, as the market for Hem Accelerate. I think it's time, and I'll hand over to Harry to talk about the next program, which is the DURA program. Harry?
Thank you, Dinesh, and thank you for your energy around Hem Accelerate. Yeah, I think we're all pretty excited about it, and the fact that we can now get it into the clinic is a very exciting time. So the DuraCAR program, many of you who have been following the company are probably familiar with it. For those who are not, we call it DuraCAR because it's a CAR T based therapy that we're developing, and we want it to be durable because we wanted to solve some of the problems in the CAR T space, and I can talk about that here. So a CAR T therapy, it's a receptor that has a part that's outside the cell, a part that's inside the cell, so here you can, on the left side, you can see the part that's outside the cell, and then it goes through the membrane, and it has a part that's inside the cell. And you can put different things on the outside and different things on the inside. There are many CAR T therapies that have been developed. Several are used in the clinic now and are treating liquid tumors. But there's a common problem. And the common problem is that the CAR T cells, when injected into a person, over time, they lose their responsiveness. And this is a problem, not necessarily for B-cell lymphomas, for example, which are being treated quite successfully, but for using these cells for solid tumors, that's one of the major problems. Now, because Regen has been focusing on this molecule called NR2F6 for quite a while, again, as you may or may not know if you follow us, and the NR2F6, these are just some papers that are out there describing NR2F6 and its role in controlling the immune system, but it's clear that NR2F6 is an immune checkpoint, and inhibiting NR2F6 activates T cells. And the idea initially was to create CAR T cells where NR2F6 is inhibited, because as you see down here, it regulates the immune checkpoints in CD4 and CD8 T cells. It inhibits the CD8 positive memory T cells. There are many types of T cells in the immune population circulating in your blood. And
hey, Eric,
yeah.
Let me jump in real quick here. So our approach to using DuraCAR seems to be widely different than the current therapies that are out there on the market. Is that a fair statement?
Absolutely.
So we're looking at solving a problem that to this point hasn't really been solved because of the diminutive returns on the therapy's viability over time. And if we can go back in and utilize our NR2F6 approach to enhance the outcome as opposed to seeing a decaying therapy, that seems to be the major benefit in the approach we're taking. Is that a fair statement?
Exactly. So we want to take the work that we've done around NR2F6 and others and take advantage of the idea of knocking out NR2F6 or increasing its level and creating different kinds of T cells that kill cancer cells and kill them durably, DuraCAR, or perhaps create T cells that are immunosuppressive because they have a lot of NR2F6. So we've been working on this now for, I don't know exactly, maybe let's call it a year or so. And we've been working with some contract research organizations, and we've been getting data back slowly, but we've been getting data back. So, so far, we've designed and validated a way to knock out NR2F6 in protein in these cells. We generated CAR T cells and CAR T cells that are expressing this new chimeric antigen receptor that I described that has this shRNA directed towards NR2F6. And now we're starting to get some of the data back where it characterizes these different CAR T cells. And so the results have been, I would say, extremely positive. So we've been able to design this shRNA NR2F6 CAR T cell, that's not a trivial thing. It was a big deal, but it's done, and we can generate them, or the contract research organization generates them for us. we are now getting back the characterization of these T cells. So we're hoping or expecting to see different markers on the T cell go up or down, and that will tell us if these cells are going to kill or are going to be immunosuppressive. And initially, we've gotten some initial data that shows that the T cells that have the right surface markers that are activated are killing tumor cells in vitro, not in animals yet, in vitro. So that is a very exciting finding. We're expecting much more comprehensive data in the coming weeks or so, and we of course will report on that. But I didn't make any of the fancy graphs or anything like that scientists love to show because we don't have all the data in yet. But I would say that we have a very, very promising data so far.
Excellent.
So as a summary, the Heme Accelerate ready for phase 1/2 studies, and the DuraCar, it's preclinical, it takes time, but it's absolutely moving in the right direction. So thank you very much.
All right, so I think I'd like to jump in and take some questions from the audience and see if we can clarify some points.
All right. Are you ready for me, David?
Yep, I'm ready for you.
Okay, great. We have a few questions from Clifford Mardson. First of all, Clifford wants to know, is the company currently actively seeking a new partnership, licensing, and funding deal with a big pharma or equivalent to help advance into phase 1 trial?
So at this point, we are exploring potential arrangements with not only big pharma and big biotech, but also looking to the investment banking community, because we know we have some money that we need to spend on research and development, but also we need to allocate funds to getting the Heme Accelerate program through clinical trials. Now, the Heme Accelerate program, because the therapy itself can be linked to treating bone marrow loss in chemotherapy patients, we're in a place where we see a very, very large market, very big opportunity in there for us that currently isn't really being tapped by other businesses. So, long and short of it, yes, we're looking for capital, and we're looking for relationships that can help move things forward.
A follow-up question, if you could give an update on the drawdown of the Coventry Enterprises equity line agreement for funding business operations?
Yeah, I think we have something less than half of the shares that we registered, which was approximately 1 million shares, a little more than 1 million shares. I think we're roughly at somewhere around 300,000 shares left. We haven't, over a month now, we haven't done any drawdowns. So that's pretty much where that sits.
Okay. And Greg Giordano says, you stated previously there was a material event that refrained you from talking about financing and partnerships. So did this event fall through? And if not, when can one anticipate it materializing?
Well, the problem you have is that you're going to read about it in an 8K when it happens. When we're negotiating with people, most of the time, the people and the types of negotiations taking place are something that isn't disclosed until the event actually has occurred. So we can't talk in real time that, hey, we're talking with XYZ company, and they're looking at doing this for us. That has potential ramifications of violating confidentiality and a number of other areas. Now, I know everybody's anxious to see what the roadmap is and where it's going. I try to be as transparent as I can be, but when I tell you I can't tell you about something, it means that there's things that preclude me from doing that. And they shouldn't be read into that it's a negative. It's an event that has to take place before it's disclosed.
We also have a question about an update on grants, more specific and definitive, when you'll be applying for a grant, how many, and for how much?
Well, I think the grants, there's another cycle coming up in, I believe it's September. So we're going to have to figure out which grants are going to be the most appropriate and focused on the areas that we're interested in, and how much is available in those grants.
And how much longer will the company have the need to dilute the RGBP common stock using the Coventry Enterprise Equity Line Agreement for funding business operations? Can you talk a little bit more about that?
Yeah, I think at the end of the day, when you're talking to a biotech company, they have voracious appetites for capital. What I'm looking at now are relationships that could be accretive as opposed to dilutive. And so in terms of what in the short run, yeah, we're going to be doing some usage of the equity line. However, as I said earlier, in the last over a month, we haven't done any equity line. So it's just a case-by-case basis. To get into clinical trials, we would like to, we prefer to find some sort of partnership arrangement as opposed to doing it ourselves through dilution and the equity line.
And Greg wants more updates on what you're doing with the science to help increase shareholder value.
Well, I think we pretty well covered that with Harry and Dinesh's contributions today. I think that, as mentioned, the chemotherapy, I think everybody out there knows that chemotherapy is widely used. And if you have a mechanism that can make it less deleterious to the patient, I think that would be a huge win. Heme Accelerate has that potential to be that piece of the puzzle that mitigates a lot of the really bad side effects of chemotherapy. I think the Duracar situation, that's moving in a very good direction and has the potential to be a major therapy. Those things, when you're talking biotechnology, you are not in an industry that you can say, well, next quarter's revenues are going to be Y, or our sales are up. You're in a highly regulated industry where the outcomes don't happen immediately.
A little bit about the products. Question about the status of your IP's license to ONPH.
ONPH had a very specific license agreement on what they were licensed for. It appears to me that ONPH is not doing business at this point in time. And they have virtually zero impact on the things that we're focused on.
And a gentleman asks, saying he thought CAR T cell were confirmed as immunosuppressant, but now you said it will be used for cancer. Can you explain that?
Yeah, of course. And,
yeah, let me lob that to Harry.
Sorry, I assumed you would. Absolutely. So in the T cells for which the NR2F6 protein is overexpressed. We expect those to be pro-autoimmune, useful in autoimmunity. And for those in which the protein levels are suppressed, which was the original idea, we would have CAR T cells, which are anti-cancer. And so the results that are starting to come in, we're getting kind of both types of cells. Remember, the T cells are a heterogeneous population of CD8, CD4, all kinds of Th17, all kinds of T cells. So we're teasing that out. But so far, it looks very positive on both sides. But you know, we need more data, which are coming.
Okay. Greg also asks, can you tell us about the arrangement you just made with ValueQuest and what their role will be regarding social media exposure?
ValueQuest is basically here to assist us in identifying usages for social media. I am not that gifted in social media platforms, and I rely on consultants to try and drag me kicking and screaming into the next millennium.
Well, we'll follow along with that. And Stephen asks or says, you've stated no more reverse splits in the future. Can you confirm that?
Well, I've already said it.
Okay.
If you're deathly afraid that I'm not telling you the truth, then get out of the stock.
Wonderful. Well, do you have any closing remarks?
Yeah. One last thought. Our issue at this point isn't having too much stock in the market. Our issue is generating liquidity so that the investors can get out of the stock or buy stock without feeling that the market's moving in a deleterious manner. Anyway. Last words from you? Last words. I'm really excited about both our DuraCAR program and the Heme Accelerate program. Heme Accelerate to me is very exciting from the standpoint that it represents our first foray into the clinical trials. And that's a real exciting place to be.
Perfect.
We appreciate the opportunity to share our thoughts and what we're doing, and look forward to having more things to communicate with you next time. Wonderful. Thank you, gentlemen, for joining us, and we'll see you in the near future. All right. Appreciate it very much, Anna. All right, everyone, stay with us. We'll be right back with Plant Beta Foods.